Morning, my name is Ricard, and today I'll share you my experience on doing single-cell multiomics data integration using multiomics factor analysis. So I don't like sharing my face during my talks, but instead I'll add you a video here on the left just to keep you entertained during my talk. Um, so when you do a single-cell experiment, at least from a computational side, what you end up with is a list of data matrices, right? So you have different data matrices on the columns that they represent different groups of cells, different donors, different batches, different conditions, and so on. At the same time, and we're starting to get not just more groups of cells, but at the same time more groups of features. So that's what we typically call different data modalities or different omics. And whenever you have to analyze this data set, you could just analyze all these matrices independently, but ideally you want to preserve this multi-group and multi-view structure of your data and do a proper integrative study of your experiment, right? So what do we mean by integrative study or data integration? And you have seen a lot of data integration methods that have been published for single-cell genomics, and they literally all have the same title, data integration for single-cell genomics. And I think this is very confusing for people that are for everyone, but specifically for people that are starting on the single-cell genomics field. So I think the first task here is to classify and contextualize these methods properly. And this is what we're trying to do with a review together with Anna Cuomo, Oliver, and John Maroney that will hopefully get published soon. But the idea is that what we classify data integration tasks in three different groups. That depends on what we call the common coordinate framework, or also the ANCOR. So for example, there are cases where you have different observations, different groups of cells, such as here. These represent different groups of cells, as I said. So what we only observe is one data modality. We observe different data matrices. That's what we call horizontal data integration. So here the features, so the genes, represent the anchor between the different data modalities. Another experimental design is when you have, from the same group of cells, you have different set of data modalities or omics. So this is represented here, right? And this is the task that we call vertical integration. So here the cells act as anchors, whereas the features are allowed to vary between the different data modalities. Now, there are more challenging tasks, such as the one here that we call diagonal integration, which is the case where you have different sets of omics profiled in different sets of cells. And here you don't have an anchor in the high dimensional space. So this integration task is significantly more challenging. And yeah, I think I'll stop here. There are different combinations of these tasks, and we discussed this in the review. But for this talk, I'll specifically focus on the vertical integration task, which is the one I've been mostly working with during my PhD. So the class of models which are extremely useful for data integration or for exploratory data analysis in general are called latent variable models. So the idea of these models is that you start with a very complex high-dimensional space, shown here on the left, and then you want to reduce the dimensionality of your space into a set of latent variables. So that's what we call the latent manifold. Now, how you do that? This depends on a function which is called f of x. This function can be defined in many different ways. It can be defined to be linear, non-linear, can be probabilistic, non-probabilistic, etc., etc. And this depends on what is the aim of your analysis, right? If you care about interpretability, which is usually the case in single-cell biology, usually this is chosen to be a linear function. And if you choose f of x to be a linear function, what this simplifies is to this matrix factorization scheme. So if this is your input data, and assuming just one data modality for simplicity, this is going to factorize into the product of two different matrices. One matrix, it's the factor matrix, so that's factors times samples, and then you have these weight matrices that what they do is they are the one relating the high dimensional space with the low dimensional space. And you see that it has dimensions features times factors. So for every feature and every factor, it provides you an importance score, right? So in here really is the key of interpretability of linear unsupervised methods. So being able to explore the weight matrices. Just as an example, methods such as PCA or factor analysis, they are just instances of this metrics factorization framework. Now, what happens if we have different views of data? So what if we have from the same set of cells, we have multi-omics data? So that's the task that we tried to solve with this model that we proposed a while back that is called multi-omics factor analysis. I won't go into the details of how this model works. I'm giving you here the references if you want to explore them. But the idea is the following. The idea is that you have your input data here shown on the left. And then you're going to reduce the dimensionality of your entire multi-omics data into a set of factors. These ones are here. Now, and critically now, the factors, some factors will be shared across all of the omics. That means that they will capture covariation across all your data modalities, but some factors will capture covariation between subsets of your data modalities or from just one data modality. So you want to disentangle which factors are capture covariation, which factors just capture variation within one data modality. The key for this is using sparsity assumptions, some sparsity priors, because this is a Bayesian framework. So we work with prior distributions, and we use this what is called the automatic relevance determination prior. And again, for details, I'll send you to the reference here. But the important thing is that once you get your factors, the same time you learn the weight matrices. And again, the weights is what relates the high dimensional space with the low dimensional space. And now you see that we have a weight matrix per data modality, right? So for every specific factor, we can see how much each feature is contributing from that specific data modality. So I'll just give you an example. Let's say that factor 1 is cell cycle. So this will be captured here. This is the first row is factor 1. And then when we look at the corresponding column on the weight matrices, you see that, for example, this gene seems to be very important for cell cycle. Same goes for gene 2, but for example, gene 3 or gene 4 doesn't have any relevance for this factor. Now, when you look at DNA methylation, you see that for DNA methylation, none of the factors are actually important for cell cycle. Whereas if you then go to chromatin accessibility, you see that cell cycle there are a lot of chromatin regions which are important for cell cycle. And then you can proceed in that way. You see that, for example, factor 5 or 6, factor 6, the one here, it's not important. It doesn't have any contribution from the RNA, but it does have a contribution from methylation, and it doesn't have any contribution from accessibility. So this is the insight that you can get with MOFA. And the most important plot that MOFA generates is the following one. So for every factor and every data modality, you get a value of variance explained. So you see that, for example, factor 1 explains 25% of the variance in this data modality and 0% of the variance in this data modality, and so on and so forth. So the reason why I like this plot so much is because it summarizes the variation from a very complex high-dimensional multi-omics dataset in a very simplistic heat map that you can easily interpret. Now, of course, you may argue factors are just abstract mathematical entities, right? You cannot really interpret what the factors are. I mean, you cannot really guess what the factors are immediately. And that's right. So the next task is to actually interpret and try to put labels on each one of your factors. So how do you do that? The easiest option is to just inspect the feature weights. So for example, if you have a good knowledge about your system, you might already spot that these genes are driving cell type X, let's say. Sometimes it gets more complex than that, so you might want to do some gene set enrichment analysis to characterize your factors. But sometimes the easiest option is to actually just plot your factors as you would do with PCA or a UMAP plot, and then you color your cells based on your covariates or your cell type definitions if you already have them before fitting the MOFA model. All right, so I think I'll stop here in terms of theory, and then I'll move straight away on how can you use this framework to analyze single-cell multiomics data. So I'll start with, I would say arguably it's the easiest multiomics experiment that is currently available, which is CiteSeq data. It's the easiest to analyze, I mean, because it has the transcriptome, which we have a very good understanding of, and then a few selected protein surface markers from the same cell. So I'm just showing here an overview of this dataset. So this dataset was published with Seurat version 3, I guess, and it's available on the Seurat webpage. So it has 2,000 genes after processing, filtering, and so on. And this is showing a UMAP of those 2,000 genes. And then it has 25 ADTs, so that's antibodies that target the surface proteins. I forgot to mention, this is derived from PBMCs. So we have around 30K cells from one healthy donor. So that's essentially blood. And cells are colored here by the cell type level, which they've done the annotation. And you can see that using both data modalities, you can sort of get most of the cell types, even if you have just 25 ADTs and 2,000 genes in here. But there are some differences. And we want to use MOFA first. We want to get a single latent space that has information from both data modalities. And then at the same time, we want to be able to quantify these differences between RNA contribution and antibody contribution. So the first plot, as I was saying, once you fit the MOFA model, is this variance decomposition analysis. So for every factor and every view, you want to get a value of variance explained. So what do we see, first of all? We see that antibody explains, so the model explains a lot more variance on the antibody data than it does on the RNA expression data. And what this means is that we could argue antibodies are less noisy, and I mean, they are targeted, right? So they are known to separate these cell types. So that's the reason why the model explains so much variance on the antibody, whereas the RNA expression data, it's more noisy and there's a lot more genes. So that's not surprising. When you look at the variance decomposition on the left, you see that most of the factors are capturing covariation between RNA and an antibody, but there are some factors which are unique to individual modalities, such as factor 6 and factor 8. So whatever factor 6 and factor 8 are capturing, it's clearly mostly driven by antibody, and there's not so much contribution from the RNA. Now we can visualize the factors. So in this case, we have cell type labeled. So what I'm doing here, I'm just plotting the factor 1. So each dot here is one cell, and then I'm stratifying and coloring by the cell type definition given by the original paper. And what you can see is that factor 1 quite clearly captures the formation of monocytes and dendritic cells compared to all the other cell types. Because that's important. Each factor is capturing variation. So you're going to have some cells at one end of the factor, and then another group of cells at the other end of the factor. So again, now you can look at the feature weights. You can visualize which are the genes and which are the antibodies that are driving factor 1. So here, each dot corresponds to one gene, and negative weight means that this gene is more highly expressed on those cells with negative factor values, whereas positive weights means that these genes are more expressed in the cells with positive factor values. And if you know PBMC biology, you know that these are clear markers of monocytes and dendritic cells. Same thing can be done with the ADTs. And you can see which surface markers are markers of each of these subpopulations here. Again, you can proceed in the same way and look at factor 3, where you see that it's capturing specifically progenitor cells. And again, you can explore feature weights and so on and so forth. Now, typically, if you have cell types definition a priori, that they come from some transfer learning approach, from some reference data set or something, you can calculate the average factor value for each cell type on each factor, and this can immediately tell you which factors are driving which different cell types. So for example, factor 9 seems to be quite specifically capturing dendritic cells, whereas factor 14 seems to be capturing plasma blast cells, and so on and so forth. There is not one correspondence between, isn't a one-to-one correspondence between cell types and factors. So one factor can be capturing different cell types, and could be also that one cell type is captured by different, by combination of factors. So sometimes when you have a lot of factors and also a lot of cell types, it becomes really hard to work in a factor by factor basis. So it gets more useful to actually combine all those factors in a non-linear way and then work on this non-linear latent representation. So then what you do is you can take the MOFA factors, you can exclude the MOFA factors which are not interesting, for example, technical variation, cell cycle variation. So you exclude all those factors because you can interpret them by looking at the feature weights, and then you build a UMAP using the MOFA factors. And you can see that you can distinguish most of these subpopulations, and then you can compare these to the UMAPs that you get with the RNA and antibody, and it should be a more complete UMAP. But of course, this largely depends on the data set. You can highlight individual factors on the UMAP space. So for example, you see that factor 1 is capturing these cells here versus all the other cells in blue. Factor 3 specifically capturing the progenitor cells that they occupy this region in the UMAP space. And there are some factors which are quite beautiful. So this is, it's not here. Actually, I'll mention this later. Okay, so having done that, the other task that we want to achieve with MOFA is we want to quantify for each one of our cell types if they are more driven by RNA or if they are more driven by antibody data. So the approach is very simple actually, and the idea is to exploit these variance decomposition analysis, but we need to tweak it slightly and recompute variance explain estimates. So instead of recomputing them by factor and view, such as the heat map I showed you before, we want to compute variance explain for each cell or for each cell type, which is just a collection of cells, and its data modality. And this is a very simple step. And then the second step is you can measure the RNA contribution for a specific cell type as the variance explained by the RNA divided by the total variance explained. So you divide RNA plus antibody. So a value of 0.5 means that RNA and antibody contribute more or less the same. And then a value higher than 0.5 means that RNA contributes more. A value less than 0.5 means that antibody contributes more to define that cell type in that MOFA latent space. So what can we see here? We can see that progenitor cells are the ones that are mostly driven by RNA, whereas mature T cells are the ones which are mostly driven by antibody data. The nice thing is that now you can calculate each one of these values per cell, so you can overlay this into your UMAP. And quite nicely, you see here that this is the progenitor subtypes. You see that they are the ones that have highest RNA contribution score, whereas the T cell subtypes, they are the ones that have the lowest RNA contribution score. So I'll finish talking about CITE-seq data now, and I'll go and talk about a slightly more complex assay, a fairly more complex assay, which is single-cell RNA-seq and single-cell ATAC-seq from the same cell. So this is the new multi-omic kit that 10x Genomics has released this year. So this work I'm showing you here, this is a collaboration that we did with the research and development team from 10x Genomics, where they tried to come up with strategies to analyze this data set. So they share with us a data set of 10K cells, again from PBMCs, from one healthy donor. And then just here, I'm showing you an overview of the data. You see that cell types are fairly similar to the CITE-seq data set. They are different because these cell annotations were done by the 10x Genomics team, and I just took them for my analysis. But keep in mind that this can be refined. So let's proceed with MOFA. Again, the first step is to define your data modalities and fit the model. Here I thought MOFA works with any number of views. So it could be interesting to try to disentangle the contribution that you have from distal peaks. So distal means that they don't overlap gene bodies that they don't overlap the transcription start site or the promoter of gene. And then on the other side, we have another data modality, which is promoter overlapping attack peaks. And then we have also RNA expression. So what can we see here? Now, when we look at the variants explained, we see that most of the variance is driven by RNA. And attack with 13 factors, we explain 10% of the variation in attack distal, for example. So that's pretty small. It's not surprising though. So if you run a PCA, for example, you see that you end up explaining similar amounts of variance. And that's simply because, attack data is way noisier than RNA data. And it's almost binary. So we need to do some normalization strategies so that it can be used for MOFA and other methods. And this limits the amount, this has an upper limit on the amount of variance that you can explain. But still, what we see is that most factors are capturing covariation between modalities, such as factor 1, 2, 3. 4 doesn't seem to be the case. 4 is mostly driven by attack. And factor 5 and 7, for example, is mostly driven by RNA. So let's proceed similar as we did before. So let's look at factor 1, and then we can stratify by cell type. And here I'm coloring cells based on whether they have a lymphoid origin or a myeloid origin. And this factor is quite clear. What is capturing is the origin of those cells, as whether they are from the lymphoid branch or the myeloid branch. And by looking at the feature weights, you can spot which are the markers that define these populations of cells. Now we can do the same thing for attack data. So you can see which are the peaks that are more enriched on cells with positive factor values in that case. That's why there's a positive sign in here. But working with peaks is quite hard because there is no interpretability in peaks. So typically what you want to do is you want to aggregate or pull information across peaks and do some sort of motif enrichment analysis. So again, you can do this and then you realize that these hits, they do make a lot of sense. Again, let's try, instead of working in a factor by factor, let's try to combine all the factors and get a UMAP out of the MOFA factors. So here you inspect your factors, you exclude the ones that represent technical variants or unwanted biological variant, and then you create your UMAP based on the remaining factors. And you'll see that, it makes sense. You get all the different subpopulations. Again, let's highlight individual factors on the UMAP space. So factor 1 is driving this population versus the other sets of cells. Factor 2 is driving this population versus the other set of cells. And quite interestingly, I like this factor a lot because it's capturing cell cycle. And you see how beautiful you have these gradients in here within each of your cell types. And I know that this is a cell cycle factor because I look at the RNA weight. And then I see that there are cell cycle genes in there. Or it's more easily in that case to do a gene set enrichment analysis. Again, let's quantify these multimodal contribution scores. And what you see here is that they are mostly distributed around 0.5. Most of them, perhaps except CD8 effector T cells, but still the value is not very extreme. So what this means is that it doesn't matter which omic are you using, that they are both contributing in a similar way to recreate that latent space. Importantly, these values are normalized by the total variance explained in here, because obviously there is more signal in the RNA. But when we normalize this, we see that actually you can get the same subpopulations. It doesn't matter whether you use the RNA or the attack. There are more genes that define them based on the RNA than the attack peaks, but you can use attack to still get the same cell type. These cell type scores, which I just introduced for the first time within the MOFA framework, they are quite. Let's say I'm still exploring them. I still have some questions on what are their statistical properties, how they behave. So if you have a data set where you want to try this, so please get in touch with me, because I think this still needs some, before officially releasing it, I want to do a bit more testing and comparisons with similar contribution score that you can get from Seurat version 4, for example. So with this, I'll finish my talk. We have a web page, which we spend quite a bit of time on. And here you can have the tutorial, installation instructions, and a lot of things that can be useful if you want to use Mofa for your own research. We have a Slack group where we are quite active. So if you have any questions, just here on the contact tab, you're going to see the address. So feel free to join and ask questions where I or others can reply. So with this, I'll finish my talk with my acknowledgement slide. So these are all the people that have contributed significantly to my research work during my PhD and now starting my postdoc at the group of Wolf Reich and as a visiting scientist at the group of Lars Belten at the CRG in Barcelona. So with this, I want to thank you for your attention